All righty. So today we're going to continue our discussion of Chapter 12, Adaptive Immunity. And specifically, we're going to go over the following topics. We're going to go over the learning objectives. I'll open it up to questions, and then we'll put everything together. We will talk about effector cell responses and how that leads to the adaptive response. Okay. Then we'll talk about types of immunity, and then we'll talk about what to do for next week. Okay, first off, this chapter helps us move toward being able to meet the following learning objectives. Being able to define the key role of evolution as it applies to microbiology, identify microbial structures and connect the structures to their functions, identify pivotal components of microbial systems important to human health, and analyze and describe the impact of microorganisms. Okay, any questions? Okay, if you think of any, let me know. I'm happy to answer them. Okay, so first off, we're going to talk about effector cells and how they lead to the adaptive immune response. And in the meantime, we will go over very, very briefly how we got to this point. Okay. So off in the bone marrow, okay, we get baby lymphocytes. And they differentiate into the point where they become CD plus 4s and CD plus 8s, okay? And B cells. Okay. B cells stay in the bone marrow. Whoops, that's supposed to be an N. Okay. Where all B cells that would potentially recognize self-antigens, okay, are eliminated. Okay, only those that recognize foreign antigens, in other words, those antigens that come from outside of the body, okay, that are not a part of the cells that are supposed to be there, okay, those get to go on. Okay. Meanwhile, our T cells, what will eventually become T cells, both of these go to the thymus. Okay. And we make sure that what leaves are those who recognize MHC, okay, not to attack it, but to be able to recognize it as a part of their job, okay, as T cells, and that they recognize, whoops, foreign non-self antigens. Okay. And these get to go on. Okay. So we're going to say that they all go on to the lymph nodes. Okay. Where they hang out waiting to find their antigen, okay, or specifically to find their epitope. Okay, which is a piece of an antigen. Okay. And what happens is we have antigen presenting cells. Okay. And so We have antigen presenting cells that show up in the lymph nodes and other lymphoid tissues. Okay, and all of our T cells, okay, gather around looking to see if the antigen that's being presented matches up. Okay. And so my CD+8s are checking out MHC1. Okay. My cells that are CD+4s are checking out the antigens that are displayed on MHC2. Okay. Meanwhile, my B cells, okay, can pull antigen from the environment, okay, an active antigen presenting cells. Okay, so my CD+4s would be looking to verify that the antigen being displayed by B cells is also foreign, and that would be displayed on MHC2s. Okay. All right, how are we doing so far? I know that's really quick. Okay, once they find their epitope, okay, then they expand. Okay, I get a whole lot more of them. Okay, so I get a lot of B cells. I get a lot of CD8 and CD4s. Okay. Now these guys, we're going to say these guys haven't found their epitope yet. So they're going to hang out and they're going to go looking, hitting the singles bars, checking out the MHC1s and the MHC2s being displayed on the dendritic cells in this case. Okay. All right. And just as a reminder, when they undergo clonal expansion, okay, they will further differentiate into memory cells. And memory cells hang out, okay, they're in the lymphatic tissue, and their job is to make more effector cells. Okay, so the effector cells for CD+8, okay, are cytotoxic T lymphocytes. Whoops, that's supposed to be now. Okay. And then the effector cells for CD+4s, we get various helper T cells. Okay. We've got helper T1, we've got helper T2, we've got helper TRs. Okay, we've got a whole slew of different jobs that your helper T cells can do. Okay, so we'll go ahead and put some of these just so that you know that their main job is to go out and talk to other cells. Okay? And then B cells, the effector cells of B cells are plasma cells. Okay? All right, how are we doing so far? You ready for me to clear the screen? We're going to talk about what happens. Okay, what do these effector cells all do? Okay. Okay, so we're going to start out with the helper T cells. Okay, and I'm going to talk about I'm going to be non-specific when it comes to the ones and the twos, because to be honest, I'm not going to test on that. Okay. But I do want you to know the difference between the ones and twos and the repressor cells. Okay. We're still learning about the repressor cells, but it's good to know what they do. Okay. So these are your CD+4 effector cells. Okay. And then we're going to have CD+8 effector cells, so your CTLs. Okay. And then we're going to talk about B cell effector cells, and those are plasma cells. Okay. All right. So coming back up here to my CD4 effector cells. Okay. So your T helper cells, whether they're 1s, 2s, 17s, okay, they go off and they inspire the troops. Okay. So they can upregulate macrophages and other phagocytic cells. Okay. So they get them to eat more. They go out, they're invigorated, they've got a package of Tums so they can overeat the pathogen. Okay, they also go talk to B cells. Okay, because remember, B cells, even if they're an antigen presenting cell, they need to have verification that the antigen that they're presenting is the antigen that we're looking for. So in other words, let's say my helper T cells have gotten confirmation from both the B cells and the macrophages that that epitope is out there. So they give that confirmation to the B cells divide, they tend to make more memory cells, more plasma cells, you get a better immune response, okay? Okay, so they also go talk to the CTLs. Okay, so they go talk to the CD+8 cells. Okay. So basically, they go around and they chat and they tell, you know, verify the rumor that's going around that this particular epitope is out there. Okay. Now, your helper T repressor cells. Okay. They help calm things down. Okay. They downregulate immune responses. Okay. So, for example, if you're exposed to an epitope that is not actually a part of a pathogen, okay, you know, like pollen, it's foreign. Okay, it can light up your immune system. Ah, it's foreign. Attack, attack, attack. Your helper T regulatory cells can say it's pollen. Don't worry about it. And that's why some people don't have seasonal allergies. At least that's what we think is going on. Okay. Also, after we've had an upregulation of an immune response, you don't want it to keep going. Okay. You don't want that to get to the point where you have an overwhelming immune response, because when you have an overwhelming immune response, you can go into septic shock, okay, where you have too much inflammation in too many parts of your body, your capillaries get too leaky, you have too much fluid leaving your circulatory system and going into your tissues, you have a drop in blood pressure, you pass out, sometimes you die. Okay. So we do want something to calm things down. Okay, and those are your HTRs. Okay. Okay, meanwhile, your plasma cells start pumping out as many antibodies as they can make. Okay. And plasma cells generally have a lifespan of about 3 days. They just wear themselves out. Okay. So these antibodies go out into the fluids of the body. Okay. Goes out into the blood, the lymph, the exudate, the interstitial fluid. Okay, so in other words, you get bathed in these lovely antibodies. Okay. So let's talk about what these antibodies do. Okay. Okay, we talked about this a bit last time, but let's go over them again. Okay, so classes of antibodies. Okay, classes, or sometimes they're referred to isotopes. Okay, that's kind of a synonym, okay, for the different kinds of antibodies. Okay, now you've got a wonderful table in your textbook, but we're going to go over more of what I'm going to be likely to test you on. Okay. So we're going to go in order of how much of this, well, no, let's go in order of, this is the first time you've experienced this epitope. So what your B cells, your plasma cells, tend to make first is IgM. Okay. Now, IgM can come in a monomer, okay? But for this class, to keep it simple, I want you to remember that it's a pentamer. Okay, it's 5 of these that are joined into this kind of snowflake appearance, okay? And they are a part of the primary or first response, okay? So the first time you've been exposed to an antigen, okay, your plasma cells are going to make IgM. And they're going to make a lot of different kinds of IgM, okay, because when you're exposed to a pathogen, you're going to excite a whole bunch of different kinds of B cells, okay? And so you're going to be making antibodies against a lot of different epitopes, okay? Because, a bacterial cell has a lot of proteins, okay? And they are all they're all going to stimulate different B cells. Okay. So in this primary response, part of what your body does over time is figure out which ones work best. Okay. Which ones actually, kill the pathogen better. Okay. Which one neutralizes the virus better. Okay. So IgM is the first one, and it's kind of, your rough draft. Okay. So if I am, say I'm doing a vaccine, okay, for somebody, and let's say 7 days later, okay, that's enough time for an adaptive immune response to show up. Okay, we're going to see a lot of IgM. Okay, and then later on, okay, we're going to see our next one, which is IgG. Okay, now when I take a blood sample, generally speaking, I'm going to see more IgG than anything else. Okay, so it's the most abundant in serum. Okay, it's also found in other fluids. Okay, but for this class, we're going to be mainly worried about the blood, the serum. Okay, the lymph, we don't take lymph samples because it would be painful for the patient, but whatever's in your serum is going to be in your lymph. Okay, so this is the most common. Okay, and we only see it as monomer. And IgG does it all. Okay, it's the best at stimulating the classical complement response. Okay, it's the best at neutralizing toxins and viruses. It's the best at clumping things. We call that agglutination. Okay, it's the best at stimulating opsonization. Okay, and not only does it do it all, okay, because this is kind of like the second draft. Generally, when plasma cells, okay, or actually the memory cells that generate the plasma cells, when they're figuring out which ones do the best job, they convert, okay, they class switch from IgM to IgG. And this is less expensive to make, so it can make more IgG than it could IgM. Okay, so that's why you get more, okay, in your fluids, but that's also part of why it does it all. Okay, is because it's kind of a refinement process. It also has to do with this FC portion, but I'm not going to make you remember that part. Okay. Also, IgG crosses the placenta. Okay, pretty much the blood of the pregnant mother and the fetus stays separate, okay, because we've got nutrient and gas exchange happening at the placental barrier, okay? But IgG is the antibody that can cross the placenta, okay? So a mother can impart immunity, okay, to her little baby before the baby is born. Because the baby is born, they haven't been exposed to anything. So it's good to have some antibodies to help knock down some pathogens so they don't get sick, okay? Otherwise, the poor little baby, you know, would get severely sick. And back in the day, we used to have a lot of babies die. And that's, you know, without the protective antibodies, okay? All right, so that's IgG. So this one comes after IgM, okay? So if I were to draw blood, say after a vaccination, and I'm looking at, say, you know, a month out, okay? Because, you know, these memory cells are still making plasma cells because, you know, 30 days out, you know, that pathogen could show up again, okay? And let's say that I, you know, this is a vaccine series where I do a booster. Okay. I don't know, I don't like COVID. I would see another bump in IgM, but at the same time, in far excess, in fact, it would be higher than that, but, you know, I didn't give myself room, I would see a lot more IgG. And I would see it showing up at the same time that I'm seeing this bump of IgM, okay? Because an awful lot of these memory cells have class switched to making IgG. So we can tell if you've had a primary exposure to an antigen or a secondary exposure to an antigen, okay? And secondary exposure, that's everything after the first exposure. Okay, how are we doing? The next most common one, okay, is IgA, okay? Now, we see IgA in all sorts of fluids, but for this class, to keep it simple, because this is probably the first time you're learning this, okay? We find this in secretions, okay? And once again, we can find this in the monomeric form, but what I want you to remember for this class is that it is a dimer when it's going to be going through the digestive tract, okay? This helps stabilize it against stomach acid, okay? So we find IgA in breast milk. Okay? So after the little baby gets born, you know, moms still want to pass immunity on, okay? So we find an awful lot of IgA in colostrum, okay, which is the first milk. It's not really milk, but yeah. And then we also find it in breast milk, and it helps coat the baby's digestive tract and protect the baby from digestive pathogens that could cause diarrhea and vomiting that could lead to dehydration. Poor little babies, we don't want them to get dehydrated. So this provides some passive immunity, okay, for babies. Okay? We also find it in tears, mucus, okay, anything that the body is oozing, if you want to think of it that way. Okay. All right. I've got to get rid of interstitial fluid here. Just get rid of all this. Okay, the next most common kind, and it is very uncommon in the blood, okay? And that is IgE. Okay. IgE is a monomer, and this Fc portion makes it so that it really likes to attach to mast cells. Okay. So mast cells, remember, they are part of the innate immune system, and they are granulocytes that hang out in tissues. Okay. And what happens is, when my antigen comes floating by and it binds to this variable region, okay, that stimulates this mast cell to dump the contents of its granules out into the environment, okay, out into the tissues where it's hanging out, okay. And so cytokines are released. Okay, so that stimulates inflammation, okay? And I bring up the specific cytokine, okay, histamines. Because IgE in countries where we don't have a lot of worm pathogens hanging out, okay, it's associated with allergies. Okay, and it's also associated with asthma that is stimulated by environmental allergens. Okay, so like folks who get asthma from breathing cigarette smoke, or get more, their asthma gets worse during the spring because they're responding to pollen. Okay, that is IgE mediated. Okay, now in areas where you do have a lot of worm infestations, we see IgE helping to control and eliminate this type of parasitic infection. Okay? And that's why we have a working hypothesis that if you're too clean, that may explain why you tend to have more allergies. And that's on a population basis. It's kind of hard to tell on an individual basis. Okay. Okay, but that's what I want you to remember for IgE. Now, the last one is IgD. We don't see that a lot. Okay. We do find them bound to the surfaces of B cells. So we think what they are shed B cell receptors. Okay. Have we figured that out for sure? Not so much, but it makes sense. Okay, questions about the different classes of antibodies? You ready to have me erase the dry erase board? You're going, no, not so much. Okay, let's talk about what antibodies do. Okay, we have talked about how, let's go ahead and, I usually do gram-positive bacteria, but just for the fun of it, let's get a little gram-negative bacterium going here that's got a whole bunch of fimbriae. Okay. All right, let's say that I have produced, and we're going to go with just IgG, just for the sake of simplicity. Okay. So I have some IgG that is specific for fimbriae. Okay. So they grab a hold of the fimbriae, okay, and so this little bacterium gets covered. Okay. Oh boy, I didn't draw that one really well. Okay, gets covered, with antibodies. So when its fimbriae is covered with antibodies, it can't grab a hold. Okay? These fimbriae are what it was going to be using to grab a hold of your cells, okay, for attachment. So what they do, the antibodies prevent attachment. Okay? The same thing happens if I'm talking about a virus, and I'm going to do non-envelope virus just because it's quicker to draw than an envelope virus. So I've got a different antibody, okay, that grabs a hold of the spikes and it prevents attachment, okay? We call this function neutralization. Okay? So it's preventing attachment. Okay, or function. Okay, because antibodies can neutralize toxins too, okay, in exoenzymes. So that's neutralization, okay? All right, now we've got all sorts of FC portions sticking up here. Oh, you know what? I just changed my mind. I am gonna make this an enveloped virus, because it's going to be easy to talk about, easier to talk about on this next part. Okay. So I've got all these FC portions. Okay. And so I've got complement. Okay. I've got complement floating around. Complement is going to bind to these FC portions. Okay. And I'm going to start the classical complement cascade. Okay. So it activates complement. Okay, through the classical pathway. Okay, so complement is going to start doing its thing. It's going to get activated. It's going to start splitting, forming enzymes that bring in other complement. Okay, and so I get max forming. And gram-negative bacteria are especially susceptible to max, okay? Because they've got two membranes, okay? They've got the outer membrane, they've got the inner membrane, they've got a thin peptidoglycan cell wall. Gram-positives, not as much, okay? Enveloped viruses, okay? You get MAC formation in the envelope, it disrupts the envelope, you disrupt the envelope, you lose the spikes, they're no longer active, okay? How are we doing so far? Okay, now because we have these complements sticking up here, okay, and also just if you have a regular, you know, antibody here, okay, even if complement has not bound to it, macrophages, okay, have receptors that allow them to grab a hold of either the FC portion of an antibody or to complement. Okay. Either way, it allows the macrophage to get a hold, okay, of the pathogen and phagocytize it. Okay. And so we call this increased ability of a macrophage or a dendrite, okay, or any other phagocytic cells to swallow down pathogens to phagocytize. We call this opsonization. Okay, so that is increased phagocytosis. Okay. All right. And I think the last one I'm going to talk about is going to be agglutination. Now, I've been drawing these guys, grabbing a hold of just one bacterium or one virus, okay? But, you know, they generally travel in packs. Okay? So if I have an antibody that's able to grab a hold here and grab a hold here, okay, I'm going to clump these guys together, okay? And so agglutination is clumping. And that makes it easier for the phagocytes to clear them on out, because if I can eat 2 or 3 or 10 bacteria in one swallow, that's a whole lot faster than picking them off one at a time. Okay. Same thing with viruses. Okay. All right, how are we doing on antibody function? Okay, now I on purpose left out the CD8s, but now I'm thinking I probably should have talked about them with T cells or T helper cells, but that's all right. We're going to get to CTL function. Okay. All right, so let's say that I have a poor little respiratory epithelial cell, okay, and we'll say it's ciliated. Okay, and it has become infected with COVID. So it's making spikes, it's starting to make capsid parts, it's making the RNA genome, and it's not really happy about it. Okay. Now, as part of the innate immune system, it will have sent out interferon saying, it's too late for me, save yourself. Okay. But also, as a part of just being a cell, okay, it's going to grab a hold of bits and pieces of everything that it's making. Okay. So it's going to grab some phospholipid proteins. Okay. So, because, you know, it's making, you know, parts to build its cell membrane. So it's going to display bits and pieces of that. You know, it makes ribosome proteins. Okay. So it's going to display bits and pieces of that, but it's also going to display bits and pieces of the virus it's making. Okay. And all of this is happening on MHC1. Because it's not an immune cell. It's not an antigen presenting dendritic cell. It's not a B cell. Okay, so it's not making MHC1. Now, meanwhile, okay, my cytotoxic T lymphocytes are patrolling because they know, okay, because a dendritic cell brought bits and pieces of COVID spike to it and it said, yep, that is the antigen I'm looking for. Okay. So what it does is it scans the MHC1 on any cell that it encounters. Okay. And if it runs across ribosome proteins, it says, nope, not my epitope. Okay. If it runs into phospholipids, okay, it says, nope, not my epitope. But if it comes here and it says, yes, this is my epitope, okay, it says, I'm sorry, you're infected. We need to have you commit suicide for the good of the rest of the body. Okay. And it does that by releasing a molecule, an enzyme called porin. And just like it sounds, it forms a pore, a channel into the cell. And it follows that up with another molecule called granuzyme. And I keep wanting to put lozyme in there, but it's granuzyme. Okay, so I think I'm closer to spelling right there. And granuzyme goes into the cell, turns on gene that causes it to undergo apoptosis. I also want to pronounce that apoptosis, but it's apoptosis. So excuse me for I've been saying it wrong. But anyway, it causes the cell to die and shrivel up. Okay. And so now that it's dead, it can't make any more parts. Okay. So it takes the virus down with it. And then in the normal course of things, macrophages will come along and say, oh yeah, you're dead. I need to clear you out. Okay, so it phagocytizes it, chops up the bits and pieces, takes it back to the lymph node and displays them to stimulate more cytotoxic T lymphocytes, okay, and helper cells. Okay, questions on CTL function? Okay, remember we call this the cell-mediated response. Okay, because we've got cells going along, scanning the surface of other cells. Okay. Now, just as a reminder, okay, natural killer cells do something similar. Okay, they also have receptors. Okay, so this one is a cytotoxic T lymphocyte. It's looking for a very specific epitope. Okay, and this one is a natural killer cell. Natural killer cells are looking for any microbial associated molecular patterns. Okay, so basically they're looking for very, very specific things, and they're looking for them and can spot them on the first exposure. Okay, these guys, we have to have a specific exposure. They've got to go through all of the business in the lymph node of having an antigen presenting cell say, yes, we're looking for you. Okay, these guys are looking for inborn signals that something is wrong, or they are looking for low levels of MHC1. Okay, either of these will cause a natural killer cell to do the same thing. It'll release porin, it'll make a pore so that the granulozyme can get in and tell the cell to undergo apoptosis. Okay, so they kind of do the same job. This one, they're looking for specific things that are inborn. Okay, so it's a part of the innate immune system. This one is looking for specific epitopes that came from exposure. And so we can make a whole lot of CTLs that are looking for lots and lots of different epitopes. Okay, whereas natural killer cells, they're never going to look for anything other than these maps. Okay, these specific patterns that we have just genetically built into us to look for. Okay. Great questions. You ready for me to ask you questions? As I try to keep myself from an evil laugh. I'm really not trying to be evil. This is good practice, okay, for the test is to practice applying what you've learned, okay? And if you don't remember or if you get it wrong, that's perfectly okay. This is practice. And when you struggle to remember something, even if you get it wrong, okay, you're going to remember the correct answer better. Okay. All right. So let me get a poll up. Okay, which of the following antibodies is found in the highest amounts in serum? I should have put which of the following isotopes of antibodies. Okay, got some good early voting. Ooh, we're at 50% already. Good job. %, 80%. Get to 100. We're almost there. We're almost there. Okay, just for the sake of time, I'm going to end the poll. Okay, the favorite answer was IgG, and you are correct. Good job. Okay. IgE, we tend to not find that in blood as much. We tend to find that stuck to the outside of cells. Okay. Specifically, mast cells. We also find it stuck to basophils. Okay. They kind of do the same job. Okay, IgDs, we actually find that in the least amounts in serum, in blood. When I get a blood sample, it's going to be really hard to find any IgD. Okay. Yay. Good job. Okay. Like I said, even if you did not get the correct answer, that's all right. We're practicing. Okay, my next question is, which of the following antibodies is found in the highest amounts in breast milk for the little itty bitty babies? Okay, we got some good early voting. Okay, we're at 70% already. 90%. Okay, for the sake of time, I'm going to end the poll. Okay, the favorite answer was IgA. Good job. That is the correct answer. Now we are going to find a little bit of IgE and we are going to find even more. You know, the second most abundant antibody in breast milk is IgG, but we were looking for the highest amounts. Okay, so IgA is the most correct answer. Okay, we're going to relaunch the poll. Okay, so which of the following antibodies is found in the highest amount on mast cells? Okay, we've got some good early voting. Ooh, we've already gotten to 50%. Okay, we stalled with 80%. Come on, we can get higher than that. Come on. There we go. Now we're at 90%. Let's see if we can get 100%. Okay, so this one was harder. It was harder. Okay, the favorite answer is C, IgE, and that is correct. Okay. Now, the next favorite answer was IgD. Okay, and I know why you picked that one is because we do find it on the surface of a cell. Okay, more than we find it floating around. And but it's B cells. We tend to find it on the surface of B cells. Okay. Mast cells, B cells, they're all cells, hard to keep them separate. Okay. And then we had also a vote for IgM. That one, so far as I know, we don't find that stuck to the outside of mast cells. Okay. That one we tend to find mainly just in serum. Okay. Nothing is 100% in biology. We're going with big picture generalizations for these questions. Okay, so let's see. Let's go ahead and relaunch the poll. I think I've got one more asking about antibodies. Yay, Okay, which of the following antibodies is found in the highest amounts as a part of a primary or first response? Okay, we've got some good early voting. Ooh, we've already passed 50%. Good job. Okay, we're at 80%. Let's see if we can get a little further. Maybe we'll stall at 80%. Let's go ahead and end the poll. Okay, far and away, the favorite was IgM. Okay, good job. We do find that in the highest amounts in a primary or first response, a first exposure to an antigen. Okay, and we did get one vote for IgG. That's not bad. We do also find some IgG, but we find it after IgM, and generally it's lesser amounts. Okay, so not as good an answer as IgM. Okay, good job. Are you done with antibodies? Are you sick to death of antibodies? By the way, hint, hint, wink, wink. This kind of thing is just made for a matching question. FYI. Okay, so let's talk about the TB test. Okay, those of us that have been in healthcare, we've had this test, okay, where they take a hypodermic and they inject just a little bit of antigen, we call it tuberculin, under your skin. It's called a subdermal injection, okay, and then they have you come back 3 days to a week later, and they measure the size of any immune response, okay? And if you have a wheal or a blister that is above a certain size, then they say that's a positive result, and they send you off for chest x-rays, looking for actual tubercles in your lungs, okay? All right, now we get to the actual question, now that I've given you some background, okay? So since we test for TB using a subdermal injection, oh, I put infection, it was supposed to be injection, excuse me, rather than drawing blood, this indicates that the adaptive immune response to TB is mainly a barrier, cell-mediated, humoral, innate, or phagocytic. Okay, we got some good early voting. We're almost to 50%. God, we're past 50%. Okay, we're at 70%. By the way, this is a hard one. I have you practice really hard application style questions in class so that when you get to the test, they don't seem as bad. Generally, the ones on the test aren't as bad. Okay, we're at 70%. We're not moving past 70%. There we go. Now we got 80%. Let's see if we got 90. Who'll give me 90? Okay, 80's going once, 80's going twice. Sold for 80%. Okay, we had a tie, okay, between barrier and phagocytic. Okay. So those were both good, well-reasoned answers. Okay, it wasn't correct, but they were well-reasoned. Okay, because we are breaking the barrier. Okay, but that is not a part of the adaptive immune response. Okay, and while phagocytosis works well with the adaptive immune response, we group it with the innate immune response. Okay, so well-reasoned answers, but incorrect. Okay. The next two favorites, okay, were cell-mediated and innate. Okay. Innate, this one's incorrect for the same reason that phagocytic and barrier were incorrect. Okay, because innate is a part of the innate immune response. Okay, So the only ones that I gave you the options for were cell-mediated and humoral. These are the only ones that are adaptive. Good job on not picking humoral, okay? Because since we're not drawing blood, we're not looking for stuff in the blood, okay? So the correct answer is cell-mediated. Okay. Yeah, mainly because unless you have one of the few that have gotten a vaccine for TB, tuberculosis, generally speaking, we have a hard time forming neutralizing antibodies, okay, to tuberculosis, to Mycobacterium tuberculosis, which is the bacterium that causes TB, okay? So what really works for your body is to mount a cell-mediated response, okay? Those cytotoxic T lymphocytes, and they recruit macrophages, okay? Macrophages also, because if you get anything as a part of the innate immune response, are going to attack anything foreign, okay? All right, so let's go ahead and relaunch the poll, and let me ask my follow-up question to the TB skin test. Okay, a positive blister is a result of okay, we've got some voting going on. Good job. And yes, this is an obnoxiously hard question. It's good to practice with obnoxiously hard questions. Ooh, we're at 90%. Let's get to 100. Oh, there we go, 100%. Good job. Okay, the favorite answer was C, inflammation at the injection site. Good job. Yes. Even though the test is to test whether you have cell-mediated response, an adapted cell-mediated response to a prior infection with tuberculosis, okay? The blister forms from inflammation at the injection site that your cytotoxic T lymphocytes help start, okay? They go around, okay? They're looking for trouble, okay? Also, whenever you get a foreign antigen into your tissues, you know, you're going to have cells of the innate immune system recognize it immediately. They're going to set off cytokines, which is going to cause dilation of those blood vessels, and we're going to have exudate, okay, leave the capillaries, which is going to cause that blister, okay? Okay, so for the other ones that are incorrect, you would think that multiplication of the antigen at the injection site would be correct, but antigen, unless it's attached to a pathogen, is not going to multiply by itself. So that's why we use antigens for vaccines, okay, is because you're not going to make more of them. You're not going to actually make the person sick unless they have a stronger innate immune response and you feel like you've got the flu. Okay. So yeah, they're not actually going to multiply, okay? You would think that it could be an allergic reaction to the TB antigens, and sometimes it is, okay? But if we have an allergic reaction, you're going to get more of a generalized rash, okay? And then if you get it too often, you can develop a hypersensitivity to it and potentially have an anaphylaxis to it, okay? Hopefully nobody ever progresses to that point, okay? This one was a reasonable answer, which is why I put it there, okay? But you're not getting friction from that injection site, okay? Not enough to raise a blister, okay? And then generally speaking, your body is really good at getting fluid that accumulates spread out, okay? Unless you're in the midst of inflammation and you want extra fluid, okay? Okay, good job no matter what you picked. Okay, I've got one more. Okay, and I can't read the question for whatever reason. Oh, there we go. What could have stimulated the inflammation? Okay, that is the positive reaction. Okay, and yes, this is combining this chapter with chapter 11, in case you haven't noticed that I was doing that. Okay, we've got some good early voting. Okay, we're almost to 50%. Oh, there we go, 50%. Okay, we're at 70%. Let's see if we can get to 100% again. Hey, we're at 80%. 90%. Let's try to get to 100. And just for the sake of time, let's go ahead and end the poll. I'm going to share results. Okay. All right. So remember, we're looking for a positive reaction. Okay. Somebody who's been exposed to TB and they still have, a response to that. So all of these things would trigger inflammation. Okay. So if you picked any of them, good job for knowing that they do trigger inflammation. Okay. But we're looking for those that are a positive, that are stimulating positive reaction, an increased blister wheel. Okay. And so we're looking for things that relate to the adaptive immune system. Okay. So that we're looking for antibodies, we're looking for CTLs. Okay. So we can eliminate these three, okay, because damaged cells, that's part of the innate immune system. You're going to have a little bit of inflammation and maybe microscopic. We may not be able to see it with our eyes. Anytime you have a break in the skin, you're going to have a little bit of inflammation, okay, just as a part of the innate immune system. So are you going to have a little bit of inflammation from any injection? Yes. Is it going to result in a positive reaction? No, it shouldn't. Same thing for macrophages, same thing for damaged cells. Okay. So if you picked complement activation stimulated by antibodies or cytokines released by CTLs, you are correct. Good job. Okay, let's go to the next one. Okay, we're getting away from that TB test. You're probably going, oh, thank goodness. We're tired of that stupid TB test series of questions. Okay. All right. So a woman has a life-threatening salmonellosis, okay, that was successfully treated with anti-salmonella. When we say anti-salmonella, it's shorthand for anti-salmonella antibodies. Okay. Why did this treatment work when antibiotics in her own immune system failed? Okay. Remember, salmonella is gram-negative. And yes, there's more than one right answer. Okay, we're at 60%. This is a harder one, huh? This requires you to think back to epidemiology and post-pathogen factors, plus the innate immune system, plus the adaptive immune system. Okay, we're at 80%. See if we can get a little higher. Okay, for the sake of time, I'm going to end the poll. Okay, the favored answer was Salmonella was introduced in larger numbers than her immune system can handle. Yes, that is a definite possibility. Yep. So going back to the chapter on epidemiology, okay, remember infectious dose. You have to have a certain number of Salmonella. And by the way, the infectious dose varies for each pathogen, okay? But you have to have a certain number of Salmonella, okay, introduced to your system to cause disease, okay? So that indicates that there was the infectious dose was reached, okay? With higher numbers than her immune system, either innate or adaptive, can handle. Probably in this case, her adaptive immune system hadn't been able to gear up yet. Okay. Okay, the next favorite answer was her strain of Salmonella replicated faster than her immune system could handle. That's also a possibility. Okay, there are some strains of Salmonella that are able to avoid the innate immune system, and so they replicate more, okay, and cause more problems. Okay. Her LPS, lipopolysaccharide, which is a virulence factor of gram-negative bacteria, remember it's a part of the outer membrane, was released as Salmonella was being killed by the antibiotics. Okay? That's also a probability. Okay, if she had a large number of Salmonella and they all start dying at once, that can lead to septic shock. Okay? And then I would also say A, which nobody picked, was also correct. Okay, because antibiotics weren't working, so it could be either the release of LPS or her strain of Salmonella was antibiotic resistant. Okay, so actually all of them are correct. Okay, so good job, whichever one you picked. Okay. All right, let's see what time we have. Oh, we have time for a few more. Let's do this one. This one's kind of fun. Let me relaunch the poll. Okay, you are in charge of making sure that employees at your health clinic receive their hepatitis B vaccination. You are also in charge of testing to confirm seroconversion. Seroconversion is what we need when you develop antibodies against a particular pathogen, okay? Either through vaccination or through actually getting the pathogen, okay? Getting hepatitis B. Recently, you have an employee that hasn't seroconverted after the third shot. Okay, for those of you that have not had the hepatitis B vaccination, it's a series of 3 shots. Okay, you administer a 4th shot. A month later, the employee still does not show antibodies against hepatitis B. Why? By the way, this does actually happen. It's rare, but it does happen. Boy, we don't have a lot of votes on this one. Oh, there we go. Now you've had time to think about it. Okay, we're at 50%. Okay, 70%. If we can get to 90% or 100% would be even better. Hey, we're almost to 100%. Okay, I think we're stalled at 90%. I'm going to go ahead and end the poll for the sake of time. Okay, the favorite answers were the employee and the carrier for hepatitis B wouldn't be able to produce neutralizing antibodies, and the employee doesn't have B cells that recognize the antigens in the subunit vaccine. Okay, so this is something that we would definitely screen for. Okay, so it is a possibility. because folks who develop chronic hepatitis B, okay, for whatever reason, they're not able to clear it out of their system. And part of that is whether they're able to produce neutralizing antibodies. Okay, they're able to produce some because they're not producing them in large numbers. Okay, so this one, yeah, you would definitely check. Okay, but this one actually is a better answer because this answer explains this one. Okay, even though, okay, in that process of making the variable region of antibodies, hepatitis B viruses do the shuffle thing to come up with as many different unique combinations as are possible. In biology, nothing is 100%, and not everybody is going to have B cells that recognize every epitope. Okay, and so very, very rarely we do see people who do not seroconvert to a vaccine. And so they don't have immunity from the vaccine. It's a little scary. Okay? And possibly if they were to get the pathogen, they would have more severe disease. Yeah. So it doesn't happen very often, but it does happen, and it's good to know that it can happen, which is why we do the blood draw after vaccination for health employees, okay, healthcare employees, to verify that they have seroconverted. Okay. All right, so we also had another answer: you haven't allowed enough time for the employee to seroconvert. Okay, that's a very, very slight possibility because generally speaking, we start seeing antibodies within 3 to 5 days. We start getting good levels, okay, closer to a month out, but we start seeing antibody levels pretty quick. Detectable, I should say, detectable levels. Okay. All right, let's see what time it is. I think we have time. No, we do not have time. But we did not get a chance to cover types of immunity, but it's pretty straightforward. And I will put these questions in the practice quiz. Okay, so you can practice thinking about natural active immunity, natural passive immunity, artificial active immunity, and artificial passive immunity. Okay. All right. So what to do for next time there? Oh, I say no assignment due on Monday or Friday. That is incorrect. The embedded tutor assignment number 3 is due on Friday. Okay, so ignore my mistake here. Okay, once you get that taken care of, your signature assignment. Oh, and I did not change that either. That's cute. This is from a year ago. All right, but your signature assignment case study is coming up. Okay, and then we're going to skip chapter 13. And we are going to move on to chapter 14, which is antibiotics. Okay. All right, let's get away from this incorrect slide that I'm going to fix right after class. Okay, questions? Any questions before we end?
So there's no poster for the signature assignment, right?
No, that
Okay.
was, we switched up the signature assignment. We did a wanted poster for a pathogen for many, many years, but we decided to switch over to a case study because, to be honest, all of the instructors were kind of sick of the wanted poster. So
I didn't see anything about a poster, but I just wanted to be sure.
no, I just didn't update it. I thought I did, but I didn't. So, and you do have the embedded tutor number 3 assignment is due this Friday.
And then really quick, just to double check, when is that signature assignment case study due?
It is due next Friday, or a week from this Friday. So it's not due this Friday, but it's due on, I think it's the 28th, if that's a Friday.
Okay, thank you.
Oh, quite well.